Good afternoon, everyone. I am Dr. Nazir Ahmed, and today we will explore an exciting topic that is gene editing in this micro teaching lesson in the subject biology or biotechnology. Before knowing gene editing, we should understand what is gene. Do you know what is gene? Anyone of you? What is gene? Every living organism, every living organism has DNA, has DNA, which is like an instruction manual made up of 4 letters: guanine, cytosine, thymine, and adenine. These 4 letters are called as nitrogenous bases. Nitrogenous bases. Bases. The letters or these letters form genes. These 4 letters form genes which control everything from skin or eye color to disease resistant in plants. In plants. So, did you know scientists can now edit these genes? These genes can remove or add to cure several life-threatening diseases by the technology that is called gene editing technology. What is gene editing method? Gene editing is a biotechnological or accurate method to change these DNA letters, allowing scientists to fix errors in mutations, and add beneficial traits or remove harmful genes. So, in simple way, gene editing is a manipulation. Manipulation is a manipulation of genetic makeup of an organism, which involves insertion, deletion, modification, or replacement of DNA in the genome of living creature. This is the definition of this gene editing. Now, question is, how does this gene editing works? Gene editing works with some tools through 3 major steps. How many steps are there? Three major steps. Step 1 is find the target gene. Step 2 is cut and modify DNA. Cut and modify DNA. Step 3 is repair, repair or change the gene. Like here, you find this double-stranded. This is the target gene. Goes to the target gene, then they cut it. They have cut this part and modify it, and again paste here in this part, modified part. So scientists supposed to locate a specific gene they want to edit using genome sequencing method. Then after they edit by editing tools, there are editing tools. They make a cut at the specific location in the DNA. Then after the cells naturally repair the break, and scientists guide the repair by insertion, deletion, or modification of genes. So this gene editing works like cut and paste method. So what are the major editing tools of this gene editing processes? There are 4 major tools. One is CRISPR-Cas9. This is CRISPR-Cas9, and it is the most popular tool. It is the most popular tool used, CRISPR-Cas9. The full form of CRISPR is clustered, regularly interspaced short palindromic repeats. This system was first identified in bacteria by the scientist Jennifer Doudna and Emanuela Charpentier, and both have been awarded a Nobel Prize in 2020. How it works, this CRISPR, how it works. This CRISPR-Cas9. This is important thing. So I will show you the slide how it works. It uses a guide RNA. This is guide RNA. Uses this guide RNA to find the target sequence. This is the target sequence. You mentioned here. This is the target sequence. DNA sequence. Okay. After that, it uses Cas9 to cut this. They have to cut it. So through this enzyme, they cut this part of DNA, allowing modifications. Then see, it is cut and desired DNA pair which you need. The desired is inserted, and this is the desired. It's a customized DNA. This is customized DNA. This is the simple process of CRISPR-Cas9. How it works. There are applications. There are several applications of this CRISPR-Cas. Like it is used for disease-resistant crops. Through this, we can develop disease-resistant crops like rice, tomatoes, bananas. We can cure genetic diseases like sickle cell anemia is there. Or it is used for cancer therapies also. Like pest control, it is used to control pests also. So these are some applications of CRISPR-Cas. The second important tool is prime editing tool. This is known as the next generation CRISPR. The next generation CRISPR. Next generation CRISPR. And it works like a word processor, word processor, like a word processor for DNA, making accurate changes. So this accurate changes without cutting both DNA strands. And now why it is better than CRISPR-9? Because it has fewer errors and side effects. Remind, fewer errors and side effects. It can fix almost any genetic mutation, and it can be used both in plants as well as animals. And its application is to fix genetic disorders and also provides drought-resistant crops. Third important tool is base editing tool. This is known for this tool is changing DNA without cutting. It is used, this technology or this tool is used for changing DNA without cutting. How it works? It directly converts one DNA letter like A. A is directly converted to T. A is directly adenine is directly converted to thymine without cutting of DNA strand. So its application is to fix single letter mutation in genetic disorders, and it is used for crop improvement, nutritional improvement in crops like higher vitamin content in vegetables. And it is used to create herbicide resistant crops. And the last one is TALENs. These are TALENs and ZFNs. These are the old but yet effective tools. They work like a CRISPR. They work like an application is like a CRISPR in these tools. So the important part is the application of this tool, of this DNA editing, gene editing technology. This method, what is its application? First and foremost application, it is used for medicine. It is used to cure diseases, used to cure several life-threatening diseases before they start. Like it holds potential in human medicine by correcting fault genes. For example, use it to cure cancers. So it is using cancer therapy, HIV treatment. This is cancer HIV treatment, and also for sickle cell diseases. And it can prevent inherited diseases before birth. It can prevent inherited diseases before birth, and also permanently cure genetic disorders. So it is used for to cure genetic disorders. Like sickle cell anemia. This is the patient which has sickle cell disease. It is cured by this CRISPR Cas9 repair mutation gene. This one, and it can improve. And this is like cancer cell with this CRISPR case to inhibit its growth. Then disease control. Fighting epidemics. So gene editing can control disease, curing insects and viruses. Like example is malaria. We can treat malaria through this technology and some virus resistances. Like other therapies, like this is the patient which has defective cells. We can remove the defective cell culture with the genetically altered therapeutic genes, then we can introduce it again. This is a process to cure these diseases. And in agriculture, this gene editing is helping to develop high yield, disease resistant, and climate resilient crops. Like if you see the history of developing agriculture, like breeding, this was the conventional breeding ways, then mutational breeding has come, then polyploidy, we find some watermelons without seeds, that is because of polyploidy. Protoplastic fusion, different species can mix and form some different transgenic breeding, and this is now the recent one technology, which is we call is gene editing technology. Through this, we can develop herbicide resistant plant using genome editing in order to help farmers control weeds. And other examples are in agriculture, like rice varieties, which is used to tolerate drought and floods, wheat to reduce gluten content, and in this tomatoes, it is used to increase. This technology is used to increase vitamin D level. Vitamin D level is increasing to boost nutrition. And in animals, farm animals like fish products, it is used to improve body mass and body height. And farm animals improve meat yield, disease resistance, environmental conservation, and protecting application. In environment, it is being explored to save endangered species and repair ecosystems. Like we have some species which are affected by climatic variation, so we can generate it through incorporated some resistant genes, and after that we can develop ecological traits to stabilize this transformation. And like we have so many extinct species. If some species are to become extinct, we can re-extend it through this technology to assisting reproduction, engineering resistance, and restoring this biodiversity. So the examples are scientists using this technology to bring back genes of extinct species and also to control invasive species. But in this gene editing method, there are some ethical concerns and risks, like unintended mutations. Like if mistakes can occur, it causes unexpected side effects. Mistakes can cause unexpected side effects. Mistakes, it's mistake. Or designer babies. Human will edit genes for traits like desirable traits like intelligence, and appearance. And biodiversity risks, like editing ecosystem must cause long-term ecological changes, and regulatory issues. Like some countries allow gene editing technology, but while other ban it. So this is the problem in this gene editing methods. But if scientists and policymakers collaborate to establish ethical guidelines for safer and responsible gene editing. So in this technology, there are numerous opportunities, numerous applications, but there are also challenges, several challenges, like target medicine. We can develop medicine for life-threatening diseases. We can develop biofuels, disease resistant, or industrial products, new industry. But it is safety is an issue, main issue. Ethical concerns are there, and regulatory challenges. So in conclusion, we come across to conclusion. So what is gene editing technology, or what is gene editing? We have known now gene editing, what is gene editing, and how it works, and what are the major tools and their uses, and also what are the applications. Now I will put one question for you. Do you think is the biggest challenge in gene editing technology today? This is for you. But in conclusion, we can say gene editing technology holds immense potential, but ethical and scientific challenges must be addressed. Thank you for listening to me. Thank you all.